where we will go over factors that affect animal diversity. Now that we've examined the evolution of animal life, we're now going to take a look at the structure of animals. We can examine an animal's body plan, which refers to the specific morphological and developmental traits that are reflected in the adult form. As stated previously, there may be many differences in adult morphology. However, these differences can usually be categorized by looking at a few distinct body plans. These body plans can differ with respect to many different aspects, such as the plane of symmetry, the tissue types involved, the body cavities that have formed, and developmental patterns. Also keep in mind that evolution is not considered to be directional. Rather, it is not working always towards an increase in complexity. Rather, it's working towards a functional relationship between an organism's body plan and structure and its ability to perform within its environment. There are two main planes of symmetry that exist in animal body plans: radial symmetry or bilateral symmetry. Please refer to the diagrams below to understand the difference between these two symmetry types. Radial symmetry is very common in sessile or planktonic animals such as corals and jellyfish. Bilateral symmetry is common in more actively moving animals such as arthropods and mammals. Also note that an absence of symmetry may also occur. This is very typical in sponges. After gastrulation occurs during embryogenesis, 2 or 3 germ layers may develop. The 3 germ layers are termed ecto, endo, and mesoderm. Ectoderm covers the overall surface of the embryo and is the outermost layer. This eventually will differentiate into the covering of the animals, or epidermis, as well as the nervous system. The endoderm is considered the innermost layer that will eventually differentiate into the lining of the digestive system, cavity, as well as organs such as the lung and liver. The mesoderm is the tissue that lays between the ectoderm and endoderm. This will eventually differentiate into muscle tissue as well as other organs. Animals can be classified depending on how many of these layers develop. Diploblastic animals only have 2 layers, the ecto and endoderm. This is common in cnidarians or jellyfish, while triploblastic animals have all 3 of these layers, ectoderm, endoderm, and mesoderm. This is common in flatworms, arthropods, as well as vertebrates. Note that there are some diploblasts that have poorly developed mesoderm. As this is poorly developed, it is not considered to be a true mesoderm, and as a result, these organisms are still classified as diploblasts. We'll now take a look at the difference between these 3 tissue types. Note the gastrulation process. This begins with the invagination of cells. Due to this invagination, we now have the formation of 2 different cell layers, the endoderm and ectoderm, as well as the chambers that reside within that organism, or the gastrula, such as the blastocele between the ecto and endoderm, and the archenteron, which opens with the blastopore to the external environment. In the presence or development of a true coelom or body cavity, we note the presence of mesoderm. In this case, this would be true mesoderm, which is the 3rd tissue that develops. Animals can also be classified according to their body cavities and types of cavities that develop. A body cavity is considered to be an air or fluid-filled space that exists between the endoderm and ectoderm. Many of these triploblastic animals have what is termed a true coelom. We would call these coelomates. This is because they have a true body cavity which is derived from the mesoderm. However, some triploblastic animals have a false cavity or a hemocoel. As a result, they are termed pseudocoelomates or fake coeloms. Their body cavity derives from mesoderm and endoderm, and thus it is not considered to be a true body cavity. These body cavities are very important for providing structural support for the animal and also to facilitate the transfer of gases, nutrients, and waste between the organism and its cells and functioning tissues. Also note that some triploblasts, such as flatworms, are considered to be acoelomates. This means that they do not have a coelom or body cavity. In addition, some other animals, such as mollusks, can have both a hemocoel or fake cavity as well as a true cavity or coelom. This diagram denotes the difference between a coelom, a hemocoel, as well as an acoelomate. Note in the true coelom, we have it being derived from the mesoderm. The endoderm resides in the middle of the animal, which then will give rise to the digestive tract. The mesoderm gives rise to the muscle tissue, which contains the body cavity, and that is surrounded by the ectoderm. In the hemocoel, however, we have a body cavity that exists between the digestive cavity or original endoderm and the mesoderm. As a result, this is not considered to be a true coelom because it does not derive only from the mesoderm. In an acoelomate, note that we do not have a body cavity. Here we have the endoderm, which leads to the development of the digestive cavity, which is immediately surrounded by the mesoderm and then the ectoderm. Animals can also be classified based on their developmental patterns. Animals can be classified as protostomes or deuterostomes based upon difference in cleavage patterns, coelom formation, and the overall fate of the blastopore. In general, protostomes have spiral determinant cleavage, while deuterostomes have radial indeterminate cleavage. In protostomes, the mesoderm will split to form a coelom, while in deuterostomes, the coelom actually forms from mesodermal cells that end up folding at the top of the archenteron, or internal cavity, of the gastrula. In protostomes, the blastopore eventually develops into the mouth, while in deuterostomes, it will develop into the anus. This chart depicts the fundamental differences described in the previous slide. If we take a look at the cleavage patterns and the differences between protostomes and deuterostomes, we note that deuterostomes have radial cleavage. This means that when these cells undergo mitosis, they do so at 90-degree angles to each other. As a result, this creates a layering effect in the dividing cells. However, protostomes have spiral cleavage. This means that the division of cells occurs in a spiral manner. This leads to a staggering in the layering of cells. With respect to coelom formation, protostomes have their coelom that develops from solid masses of mesoderm that split onto either side of the developing archenteron. Deuterostomes, on the other hand, have a mass of mesodermal cells that reside at the top of the archenteron. This will eventually fold to form the coelom. And finally, with respect to the overall fate of the blastopore, in the development of protostomes, the blastopore will eventually form the mouth, while the anus will then form as the archenteron invaginates and then meets with the top of the epidermis or the ectodermal cells. However, in deuterostomes, the blastopore will eventually form the anus, whereas as the digestive tube develops and the top of the archenteron meets the ectoderm, in deuterostomes this will eventually form the mouth. Today we have 35 extant phyla of animals. Many of these animals can have their origins traced in the fossil record to approximately 500 million years ago. We consider animals both extinct and extant to be a monophyletic group. This means they share a common ancestor and they form a clade that we can call metazoa. We can also refer to these organisms as metazoans. Sponges belong to the phylum Porifera. Sponges are considered to be a basal group that actually form a sister group to modern-day animals. Eumetazoans are used to refer to as true animals. This includes all other animals other than sponges that have true tissues, such as mesoderm, ectoderm, and endoderm. This also includes diploblasts with radial symmetry. Along those lines of the planes of symmetry, most of the modern-day animals belong to the clade Bilateria, meaning that they have bilateral planes of symmetry. This includes many of the triploblastic animals that contain all three germ layers. Today, animals can be represented by three major clades: Deuterostomia, Lophotrochozoa, and Ecdysozoa. This slide represents the phylogenetic relationship between all animals. Note that the term metazoa can be used to refer to Porifera as well as all other animals. Eumetazoans, however, refer to animals and excludes the phylum Porifera, or sponges. Also note the development of bilateria and then the overall classification of the deuterostomes, lophotrochozoans, as well as ecdysozoans. When examining the phylogeny of animals, it is important to remember a few key points. Though we can state that animals that are classified within the clade Deuterostomia all have deuterostome development, this does not represent the fact that there are some other organisms that have been grouped in other lineages that also have deuterostome development. Thus, this clade is not all-inclusive to all deuterostomes. For instance, some brachiopods are known to have protostome, deuterostome, or even intermediate modes of development. Another important point to consider is the fact that hemichordates share many characteristics with chordates, such as gill slits and a dorsal nerve cord, that are not present in echinoderms. In this instance, this is an example of some of these derived traits that have been secondarily lost in evolution. A few more important points to consider is the fact that invertebrates arise in multiple lineages. Invertebrates are entirely represented in the lophotrochozoan and ecdysozoan clades. However, invertebrates also arise within the phylum Chordata, although the phylum Chordata will also include vertebrate groups. When we consider the lophotrochozoans, they are named based on the presence of two distinct characteristics. They have a unique larval stage that is called the trochophore, and in addition, they have a lophophore present in the adult stage. The lophophore refers to a ring of ciliated tentacles that aid in the feeding process. The lophotrochozoans will have either one or both of these characteristics present. Ecdysozoans are considered to be unique due to their ability to secrete an exoskeleton. This exoskeleton needs to be shed each time the organism grows or molts. This is done through a process called ecdysis. However, keep in mind that these are not the only organisms that can undergo ecdysis or molting. There are some taxa, such as leeches, that are known to molt and shed their cuticle. However, they are not considered to be part of this clade. This can be evident in the medicinal leech Hirudo medicinalis. It will molt and shed its cuticle as it progresses through its nymphal stages. However, it is not considered to be an ecdysozoan. However, insects like this cicada can leave behind their shell of the previous form as they molt into the adult stage. And finally, also keep in mind that our understanding of these relationships is constantly evolving. It is currently debated whether or not the ctenophores are considered to be a basal metazoan. This is because they have metazoan characteristics. However, they lack cells that resemble the choanoflagellates that we commonly see in the sponges. It is also debated whether the phylum Aciella, which contain the acilomates, are considered to be a basal bilaterian or whether or not they should be placed in with the rest of the deuterostomes. It is originally believed that bilaterians have descended from a common flatworm-like ancestor like the acilomates. This ancestor would have had a simple nervous system, sac-like gut, one opening, and no excretory system, once again similar to the acilomates. Thus, as our understanding of these organisms evolve, and the understanding of their relationships between the two, and the ability for us to discover these organisms as well as perform molecular analysis on them, we are constantly resolving and reevaluating the phylogenetic relationships that exist.